Hello again, students. So those of you who just finished watching the um, Frozen, the sort of basic Frozen lecture, now we need to start talking about Frozen sections that have special stains done on them and the protocols and procedures, steps, functions, and results for those. I'll also give you examples of what they will look like microscopically. And remember that we are doing these on frozen sections. However, many of these can also be done on paraffin embedded sections. And so always sort of keep that in mind. If it's a frozen section, you will not do the deparaffin hydrate step. Yeah, if it is a paraffin embedded section, yeah, then you will do the deparaffin hydrate to water and start the stain from there. So the first one that we're going to talk about is the oil red O. Now, the oil red O is only done on frozen sections. And that's because when we go through processing, yeah, the lipids or the fats are washed away. And so, yeah, we do an oil red O when we are actually demonstrating neutral lipids. And it's usually a fat content study, yeah, to see how much true fat is there. Yeah, so we're demonstrating neutral lipids. Yeah, so you're going to collect your frozen section. Then you're going to fix it in a 10% NBF. Yeah, now with your normal H&Es, yeah, your fixation is an 80% fixation for your H&E. Yeah, but with the oil red O, you need to fix it in 10% neutral buffered formalin. So at your frozen section stain line, you're just going to have a little Copeland jar of NBF. Yeah, you should never, ever during this process use any type of alcohol. This is why we fix in the NBF rather than in the 80% alcohol. Because if you go in that 80% alcohol, you're going to wash away your fats, your lipids. So your fixative is 10% NBF. You, as with any frozen section, whether it's an H&E or whether it's a special stain that you're doing, you should always be using charged slides. Yeah, that way you avoid your section washing off. Now, when we do a fat study on a frozen section, it's really based on each individual lab as to whether you need to use a control or not. The CAP doesn't say we have to. But the reason for that is because most tissues contain fat in the first place. So the patient can actually be used as your control. Not to mention that it's really difficult for frozen sectioning to have controls on hand, because we have to have the same type of slide. We would need, if we were going to use a control, we would need to have pre-cut frozen sections and keep them frozen. Yeah, so really the only way that we can do that is by having another holding mechanism like a -20-degree freezer that we would hold those slides in. And then after you collected the patient, you could go and pull one of those slides and run them together. With the oil red O, because it is fat demonstrated, yeah, we are going to mount with aqueous media. And so that makes this particular slide not permanent. So the oil red O sort of needs to be read by your pathologist pretty quickly because it's aqueous, and that aqueous media is going to evaporate out. So this is not a slide that we can hold in the file after we're done reading it. The pathologist will issue a result on it, and then the slide is basically, trashed because we can't hold it. Now, tricky. Fat is a liquid. Yeah, so when you cover slip, yeah, you cannot put any pressure on that cover slip. So you can, come out of your water and you're just gonna bloop, mount it, wipe the back of it, and be done with it. Don't chase any bubbles. Don't drop your slide onto the cover slip. Make sure the cover slip comes up to your slide and sort of runs, the water runs in between the slide, yeah, and the cover slip. And when you pack your slide over to give it to the pathologist, make sure that you're keeping your slide level. If you're tilting your slide or you're dunking your slide like this in your solutions, you are displacing that fat. So the fat has the ability to move, and it's called squishy fat. Yeah, so you have to be really careful with your slide when you're moving it from one solution to the other. It's a nice gentle movement. Do not dunky, and don't squish your cover slip onto the section itself when you're mounting. All right, so our protocol. The first thing we're going to do is go into a 10% NBF, or, you can use straight up formaldehyde. Doesn't really matter. Yeah, either one of them are going to fix your tissue. Then you're going to go into a water wash. So again, you're going to have this stain line of Copeland jars. You're just going to move your slide from one and drop it into the next. Okay, no dunky. So you're going to pull your slide out of your formalin and you're going to drop it into that water. You're just going to let it soak there for a little while to get that formalin off. Now, you don't want to drag that formalin, so this is another water wash where you need to make sure that when you're done with it, you put fresh water there, okay? From there, and you're going to soak until, you can gently pull your slide up and you can see that the formalin has come off of your slide. Then you're going to move it over and you're going to drop it into your oil red O stain. It's a Copeland jar of oil red O. And you're just going to leave it there. This is going to be your fat stain. The oil red O is red, and so it's going to stain your lipids a reddy sort of orangey color. Ours kind of comes out more orange than on the red side. And that's okay, as long as you're getting some penetration of that fat to be seen microscopically. So then you're going to pull out of that oil red O and you're going to drop into another water. And that's just going to, remove that excess oil red O off of your slide. And then gently move it out of your water and put it into a Harris hematoxylin. Now, Carson's going to say that you need a Harris hematoxylin with acetic acid. Most hematoxylins, most manufactured hematoxylins already have acetic acid in them. So you really don't have to worry about it. Yeah, but if you want your hematoxylin to absorb quicker, you can just take a transfer pipette of acetic acid and put like 1 or 2 drops in a Copeland jar, and it will absorb quicker for you. So that's your hematoxylin is your nuclear stain, okay? And then once you're done with that nuclear stain, again, you're dealing with the frozen section. So things are going to absorb quicker. So, we're eyeballing this to see that depth. Now, once you are done with that hematoxylin, then you're going to go into a water wash. Now, this is going to remove that excess hematoxylin just off of your slide. Yeah, you can, this step can be used as if you leave it in the water, and just let it soak in the water for a long time. You can use it as a bluing step, but be careful because leaving the section in the water to blue, you're actually, because you got to leave it there for quite some time, you're actually taking the chance of your section washing off. So I will put it in my water and then I'll pull it from my water and I will move it into a blue for just a few seconds. And then move it into another thing of water to stop that bluing. That water right there is your last step. So you're going to come out of that bluing and then you're just simply going to mount it utilizing the water that is on the slide. And then you can take a look at it microscopically. So this is what it's going to look like. Your fats are going to be red from the oil red O. Again, ours is kind of more on the orange side. And then your nuclei and any other elements are going to be purple from the hematoxylin. Then we have the Sudan black B. So this is also staining neutral lipids. Again, you're going to use a 10% fixative, because we don't want to use any alcohol to wash away our fats. Same story, same song and dance. Always use a charged slide. You can or cannot use a control. You mount with aqueous media and never forget that fat's a liquid and can be displaced. So notes about your Sudan black B. The Sudan black B stain itself does not impregnate the lipid. So what happens is we have to coat the lipid with a propylene glycol, and it's going to attach to the lipid cell. And then we're going to stain the propylene glycol with the Sudan black B. So if the yellow is your lipid, we're going to coat that lipid with propylene glycol. And then the Sudan black B is going to attach to that propylene glycol. So our protocol, you're going to fix in a 10% formalin or 37 to 40% formaldehyde. Then you're going to water wash to remove that. Then you're going to go into a 100% propylene glycol. This is that introduction of glycol, and it's going to coat your lipids for you. From there, you're just going to go straight into Sudan black B. This is your lipid stain. It's propylene glycol based. That's why we had to sort of introduce that glycol. And it's the Sudan black B is going to attach to the 100% glycol that is attached to the lipids. From there, you're going to move into a 95% propylene glycol. Now, this is the differentiation. This is what's going to pull the Sudan black B out of the other elements. And it's really not the propylene glycol itself that's doing the differentiation, but rather it's the 15% water that is loosening the Sudan black B from all the other elements. Then you go into a water wash. This is where it's going to continue to differentiate out of all of the other elements, leaving behind your lipid that is coated with the 100% propylene that now the Sudan black B is attached to. And this water wash is going to remove all that excess 85% propylene glycol while continuing to differentiate and pull the Sudan black B out of all of the other elements. From there, you're going to move into NFR. NFR is nuclear fast red. This is the counter stain to the Sudan Black Bee, and it's a nuclear stain. Then from there, you're going to go into a water wash. And that water wash is going to remove all the excess off of the slide, all the excess NFR off of the slide. And then you're going to come out of that water wash and you're simply going to put a cover slip on it. And this is what it's going to look like. So your fats, depending on the purity of your Sudan Black Bee, if it's not extremely pure, your fats are going to be kind of a purpley color, bluey purpley color. If it is pure, it's going to be nice and black, and then your nuclei are going to be red. Then we have the Cahals. Now, the Cahals has been replaced with immunohistochemistry, mainly because the Cahals is an extremely tricky stain to perform. So when we are doing the Cahals, the chemicals in the Cahals has to be as pure as possible. Yeah, if they are not pure, you're going to get all of these impurities interfering with your astrocytes, and that's what we are staining for. So this is a stain that can be ran on formalin tissue, formalin fixed tissue. But if for whatever reason you have fixed in formalin ammonium bromide or in formalin, then you got to place your section in formalin ammonium bromide for 2 days to sort of release that ammonium out of the tissue. So you're going to do a frozen section, and this is really tricky. I have only done this once in my entire career, and it semi-worked, but it wasn't perfected. And therefore, we switched to immunohistochemistries to stain our astrocytes. So this is done on what's called a free floating section. So you are not using a slide. You have cut a frozen, and you are using the section only. And you have this little glass rod that you roll the section up onto. And then you have what are called watch glasses. And it's just like, if you had a watch on and you took the glass off the top of it and you flipped it upside down, it's just this little bowl, okay? This little glass bowl. So you have a glass rod that you have collected your section, and then it's like sort of wrapped around this little glass rod. And then you have your watch glasses lined up. So the first thing that you do is you take your rod and you stick it down into a watch glass that has water in it, and you unroll it and you release that section off of the glass rod. And then it's going to float on the surface of that water. And you can kind of take your watch glass and sort of jiggle it a little bit, and it'll that section will move back and forth. Then you take your glass rod and you go back into your watch glass and you carefully just sort of wrap that section around that glass rod and you move to the next one. Or you can gently pour the water out of the watch glass and then refill it with your next solution. So the next solution is your gold sublimate. And you're going to let that, take that to the refrigerator and let it sit in the dark, okay? Your gold sublimate is your astrocytic feet. It's going to stain the little feet of the astrocytes. So that's the actual stain for your astrocytes. So you're going to leave it in the dark. I left mine overnight. And then you come back and you pull that out. You sort of pour that gold sublimate out, and on the other side, you're pouring a little bit of water in, or you can use your glass rod to move it into another watch glass. At first, I picked up my section with the glass rod and I put it in my water. And then from there, I would pour it out and put the new solution in it, and then pour it out and put the new solution in it. The trick to pouring it out is that you want to make sure that you always leave a little bit in the bottom of the watch glass to keep that section floating so that it doesn't attach to the bottom of the watch glass, and then sort of keep pouring underneath of it until you think it comes clean. So I left mine in the refrigerator overnight, came out, poured the gold sublimate out, poured some water into it, sort of jiggled it, let the specimen, sort of wash back and forth on the surface of that water. And then you pour that water out and you put sodium thiosulfate in it. Sodium thiosulfate, you're going to learn, is your friend. It goes in and it cleans up all sorts of unreacted silvers or gold for you. It will clean up what's sodium thiosulfate will go into a stain and it will attach to a lot of things because it's hypotonic. So it's going to go in and attach to anything that's unbound. And then when you water wash afterwards, it'll wash away that sodium thiosulfate that's attached to anything that's unbound. So you're going to learn that sodium thiosulfate, also called hypo, is going to be your friend. So it's going to attach to any unbound gold sublimate. And then when you go into the next water wash, you pour the hypo out, you bring the water in. When you do that hypo is going to wash away. Then you're just going to mount your section onto a slide. And at this point, you are going to have to, because this is a permanent stain, you are going, because you mount it on the slide, now you're going to have to really gently, sort of just give it a little bit of alcohol, give it a little bit of xylene, and then you're going to mount it. So your results, your astrocytes are going to be black. That's all there is to it. So you can see, because of the trickiness of this stain, why it's been replaced by IHC. Okay. This is what it looks like. So depending upon the purity of your gold sublimate, most of the time it's going to look like this. These are your astrocytes with the little astrocytic feet sticking out from them, and they are wrapped around the myelin sheath. So your little astrocytic feet are down here, and that's what they're looking for, to see how many astrocytes are actually truly attached to that myelin sheath. So this and this and this, this and this are your normal Cajal stain. This is a Cajals that has been countered with an NFR. This and this are a Cajals, but these are what, this is an impure dye. So you sort of have this yellow background that, and you see the astrocyte isn't really black. This is more what mine looked like, or more like this, which is an impure dye. We would rather have it look something like this, where we can literally see those feet attached to the myelin sheath. Then we have the PAS. So the PAS stands for periodic acid shifts. There are many variations of the PAS. So the PAS will stain polysaccharides. It will stain neutral mucose substances. It will stain basement membranes, and it will stain fungus. So there's sort of this general PAS protocol. And then after you've ran that general PAS protocol, it varies based on what you've done to that stain afterwards. So if you look, oh, flashing. If you look right here, here's your general PAS protocol. If you counter the PAS with a hematoxylin, then the stain name becomes PASH. If you counter your PAS with light green, and that is for fungus, then it becomes a PASG, or some people will call it a PASF. So the F stands for PAS fungus. The G stands for PAS green, which is fungus as well. It just depends on sort of your lab and who calls it what. Okay, but it's the same stain. Same stain, PASG, PASF, same stain. Then we have another one that's called the PAS Alcian Blue. So it's the PAS Alk Blue. This is what is referred to as a piggyback stain, because the Alcian Blue is a stain in and of itself, and the PAS is a stain in and of itself. So it's two special stains that are attached together. And we'll get there. We're going to go through the PASH, the PASG, and the PAS with diastase, but we're going to leave alone the PAS Alk Blue for now. The PAS with diastase is a PASH, except that we have digested out the glycogen in the slide. So it's a comparison, between I'm digested and I am not digested, so that we can see the true amount of glycogen in the patient. And we're going to walk through these. So your general PAS protocol. You're going to depair and hydrate to water if it's paraffin. If not, you are going to fix it in an MBF, and water wash. Then you're going to go into periodic acid. Periodic acid is an oxidation step, so we're creating that aldehyde, okay? Now, in a pinch, if you do not have periodic acid for some reason or another, your shipment didn't come in, somebody forgot to order it, I'm talking in a pinch. It is preferred that you use periodic acid, but in a pinch, a trick is that you can also use chromic acid. But if you do use chromic acid is a much stronger oxidizer than periodic acid. So you can actually over-oxidate with chromic acid. So if you do use chromic acid, then you need to significantly decrease the amount of time that you are in your oxidation step. Not to mention, you need to increase the water wash afterwards, because chromic acid is an orange color, and that orange color will remain in your tissue. So when you wash afterwards, you have to wash that section long enough so that the orange coloration is removed completely from your tissue, until you go into shifts. Otherwise, you're going to obscure your whole stain. So again, the periodic acid is preferred, but in a pinch, you can use chromic acid. So you're going to water wash, and that's going to remove that periodic acid and stop the oxidation process from happening. From there, you're going to go into Schiff's reagent. So Schiff's lives in the refrigerator. It is a pinky straw color, and you know, it does not show up as a color in your section until you hit the water afterwards. That water afterwards is what's called a development step. And that's going to bring this really faint pink color, up to a bright pink color. So you're going to go in your Schiff's. Your Schiff's is basic Fuchsin based. That's the dye component that's in Schiff's. And it's for your glycogen. Okay, it's going to stain your glycogen. Now, there's this optional step. You can go straight from Schiff's into metabisulfite, and that's going to remove any non-specific staining that you may have with your Schiff's. This is an optional step. For me, I never did it, because to me, it's kind of a lazy step, if you will. If you have worked up your PAS protocol appropriately in the first place, then you shouldn't have to go into a metabisulfite. But many labs will do it, so just keep that in mind. So what will happen if you're using the metabisulfite? You'll come out of your Schiff's, you'll go directly into metabisulfite. It's going to attach to any Schiff's that is not attached to anything else. And then when you water wash afterwards, that's going to wash away. If you and develop up that pink color from your Schiff's. If you don't use your metabisulfite, you're just going to go from your Schiff's into that water wash, to remove the excess Schiff's and to bring that pink color up. Okay, that is your basic PAS. From this point, from this water right here, from this water is where the protocol changes, okay? So if we are staining for glycogen, specifically glycogen, basement membranes, and polysaccharides, which is glycogen, you're going to be doing a PASH, which simply means you're going to tag it with a hematoxylin. So you're going to run that PAS protocol. You're going to run this protocol. From this water, you're going to go into a Harris hematoxylin. And then the Harris hematoxylin, is going to be your nuclear stain. So then you're going to water wash. We're going progressive. So you're going to water wash. That's going to remove your excess hematoxylin. You're going to go into bluing to blue your hematoxylin. Then you're going to water wash to stop your bluing, and you're going to DCM. So your PASH results, your glycogen in your basement membranes, are going to be a rosy color, a pink rosy color, and your nuclei are going to be that purple blue. And this is what it looks like. So you have your basement membranes, these nice, beautiful basement membranes. By the way, with the PASH, your control is kidney, because most of the time we're running a PASH on a kidney patient in the first place. And your kidney has all this beautiful basement membrane around the glomeruli. So there's your positivity with the shifts, and then you have your purple nuclei. Now, should your patient actually have a fungal component within their tissue, the shifts will stain that fungus as well. It's just that we didn't know the fungus was there, right? Yeah, so we run a PASH and sort of randomly figure out that this person has fungus. Your fungus is going to turn up pink. So this is fungus. This is filamentous fungus. Yeah, that has been sort of arbitrarily found in a PASH. Now, the PASG, yeah, take note that fungus is a microorganism. So there are 3 times that you have to use distilled water. And that's when you're running microorganisms, when you're doing silver stains, and when you're doing immunohistochemistries. So if we are indeed doing a PASG, then your PAS, that original PAS protocol, you should be using distilled water in that protocol, just so that you're not introducing a contaminant fungus from the tap water and giving your patient a false positive. So you've used distilled water through your PAS protocol. You come out of that last distilled water. One more right here. Okay. So this needs to be distilled water and this needs to be distilled water. Yeah. So you're coming out of this last distilled water. Then you're going to go into a light or fast green. Different name, same thing. Just a little bit different makeup of the solution. Okay. This is going to be what's called it's a counter stain, but it truly is a background stain. It's called a background stain, as opposed to a counter. A counter stain is when you have a true target, like with the PASH, our true target was the nuclei. When we're using light green, it really doesn't have a target. It's just going to stain everything that's left over. So it's kind of this blanket stain. And a blanket stain is technically referred to as a background stain, not a counter stain. So yeah, you're going to hit it with the light green. And keep in mind that water will remove your light green. So when you hit this water right here, you need to sort of keep your eyes on it while you're washing it, so that you don't fade away all of your light green. If you do pull all of your light green, kind of a no harm, no foul, as long as you realize that you did it. And you can go right back into light green and restain with it, and then go back to water. After you're done with your light green, you're gonna DCM, but guess what? Your alcohols are also going to fade your green. So you kind of have to, as you're DCMing, you sort of have to keep an eye on that green. And as long as you haven't hit xylene, the minute you hit xylene, that's what your stain is. Okay. But if you're going through those 95s and those 100s and your green starts to really lighten up, just back right up and go into water and then go back to your light green, stain it again, and then go forwards. This is what it will look like. Yeah, so these right here are budding fungus and they are stained with the shifts. Yeah, and then everything else is stained with a light green. Then we have the PAS with digestion. So this is a PASH, okay? The stain itself is a PASH. The difference comes in the very beginning of the protocol. And that is that after you have depaired and hydrated, or after you have fixed and gone through water wash with the frozen, you need to put a digestion enzyme on the slide. And you're actually going to have two slides. You're going to have two patient slides, one with digestion and one without digestion, because this is that comparison. We want to see exactly how much glycogen is in that patient. So the pathologist is going to compare these slides against each other. So one slide is going to be just a PASH. The other slide, and that would be without. Okay, so you're going to put a WO on this slide. It would be without digestion. This slide is with digestion. So when you come out of that water, you need to put an enzyme on this slide that's going to digest out the glycogen. Now that enzyme could be amylase, or it can be human spit. In the old days, we used to just spit on the slide, and leave our spit on the slide to digest, because you have enzymes, that digestive enzymes in your mouth. That's what your spit is. So we used to just spit on the slide to use that as digestion. So oh, guess we just did that. This is what it will look like. So this is a PAS without digestion. This is the PAS with digestion. So the one that has all the glycogen in it is the one that has not been digested. So you can see all the pink glycogen staining. And then this is the one where the glycogen has been digested out. Then we have the Gro-Kots methenamine silver. This is referred to as GMS. Yeah, you're going to see Gro-Kots a lot, because this guy is, he's developed a lot of stains. Yeah, so don't let that throw you. Yeah, the GMS is demonstrating fungus, and it's being done. It's a silver nitrate stain, so we're going to use distilled water throughout the whole process. We're going to oxidize the fungal cell walls using an aldehyde, creating an aldehyde, right, with chromic acid. That way, the staining can occur when it's called reduction, through the reduction process of silver impregnation. And that silver impregnation is going to be done with silver nitrate, and you're going to warm it up in the microwave. And reduction means we're going to take the silver color that's in the silver nitrate and we are going to reduce it to a visible black metallic color. Okay, that's what that reduction means. So it can be ran on both frozen or embedded. And either way with this one, you have to use a control. And the control that you use has to be a known positive fungus control. It can be any tissue type you want, but we have to know that in that tissue there is fungus. And again, distilled water, right? So the 3 times, I'll reiterate it again, the 3 times you have to use distilled water: immunohistochemistries, microorganisms, and silver stains. And this stain is 2 of those. It's a microorganism and a silver stain. So you have to use distilled water. So our protocol, you're either going to depair and hydrate, or you're going to fix in a 10% NBF and water wash. Then you're going to go into chromic acid. Chromic acid is our oxidation. So it's going to create that aldehyde for us to use. Then we're going to go into distilled water. That's going to remove that, stop that oxidation process from happening and remove that chromic acid, which will leave behind that orangey color I talked about earlier. So you have to water wash long enough to remove the orange, or it's going to interfere with your subsequent staining. Then you're going to go into a 1% sodium bisulfite. This is sort of our safety net. This is going to attach to any of that chromic acid that may be left, and avoid the non-specific staining that can occur from that. Then you're going to go into another distilled water, and that's going to remove that bisulfite that's attached to any unbound chromic acid. Then you're going to go into your silver nitrate. So this is what's referred to as impregnation. And the impregnation is done by heating up the silver. So you will do this in the microwave. And so you get a glass boat, and you have your silver in a Copeland jar. You'll put your slide in the Copeland jar, and you'll put it in the microwave. I always go like, 40%, 50%, for maybe 30 seconds, and then I'll pull it out and look at it. And then I'll go back in and do it again and pull it out and look at it. To make sure that you are not overheating that silver. If you overheat that silver, you're going to do this thing that's called flashing. And so what will happen is the Copeland jar and your slide and your tissue on your slide will turn into a mirror. In fact, that's exactly how they make mirrors for your bathroom. Any mirror is made with silver by flashing silver nitrate onto glass. So you don't want to heat up your silver too quickly, and you don't want to leave your section in the silver for too long. And it's okay to like warm up your silver, and then pull it out and let it sit on the counter and let it sort of impregnate, for a longer period of time. Then you're going to go into a distilled water wash, and that is going to remove the excess silver. Then you're going to do what's called toning. So this is a toggle step. And so you're in water right now, right? You've come out of your silver, you've put your slide into a Copeland jar of water. You need to give it a nice rinse. Dump it out a few times, clean that water. So you put water in it, you dump it out, you put water in it, you dump it out, you put water in it, and you dump it out. Um, then you're going to go into your gold. Now your gold is going to bring down, this is your reduction step. It's called toning. It's going to bring down that metal color to a black color. After that, you're going to go into a distilled water wash. So when you hit that distilled water wash, you need to keep your toning, but your toning solution, but you're going to stop the toning from happening. Then you're going to pull that out and you're going to put it underneath the microscope, and you're going to see if your fungi, and we're talking the control here, if your fungi are black. If your fungi are black, great. Stop right there. Drop back in the water with your patient. If your fungi are like a light brown, okay, you need to probably, if it's sort of a silvery brown color, you probably need to take a couple more dips in your gold, okay, and then go back into the water and check it microscopically. If your fungi are red, you have overtoned. Meaning you've gone too far, you've reduced it too far, and subsequently it won't withstand the rest of your stain process because you have actually detached the aldehyde. So you're done at that point. You have to start the whole stain over. Okay. So little bits at a time. Tone for a little bit, water wash, check it microscopically. Tone for a little bit, water wash, check it microscopically. Now, something else that you have to consider if you do go back to toning is that there is now gold chloride in that next water wash. So you have to be dumping that water wash and getting fresh water all the time when you're toggling, okay? Otherwise, toning is going to continue to happen. Yeah, so keep that in mind. Now, once you're done with your toning step, then you're going to go into our friend, sodium thiosulfate. So sodium thiosulfate, or hypo, is going to go in. It's going to attach to any unreacted silver. And then in the distilled water wash afterwards, the hypo that is attached to the unreacted silver is going to wash away. Then we're going to background stain. That blanket background stain is light green again. And remember that water and alcohols will pull that light green, so you need to keep eyes on it when you go into the water wash afterwards. And that water wash is going to remove any excess green off of your slide. And then you're going to DCM. So your fungus should be black. Your background should be green. And if you have any mucins that are there, if they're present, they will be sort of a taupey gray color. And this is what it looks like. So this is your filamentous fungus. And then everything in the background is green. This is budding fungus, same GMS procedure, but this is budding fungus, filamentous fungus. Oops, filamentous fungus is long. It's actually, one of the best known ones is athlete's foot. It's long and it has the septa, and sort of stretches out through the tissue, where budding fungus, actually buds into chains and breaks apart. So this is what the GMS looks like with budding fungus. Then we have the PAMS. So tells you what it is in the name, right? A lot of them tell you what it is in the name. Gro-Kotz methenamine silver. It's a methenamine silver, right? PAS, periodic acid shifts. Same situation here, PAMS, periodic acid methenamine silver. So we're going to discuss this guy, yeah, in the carbon amyloid lecture. But for now, you sort of need to know the differences in the oxidations. So your PAS oxidizes with periodic acid. That's what the PA stands for. Okay. Your PAMS is oxidized with periodic acid. That's what this PA stands for, okay? And then your GMS uses chromic acid. So just kind of embed that into your mind. And we'll talk about that PAMS when we get to carbon amyloids. So some few GMS tidbits. Again, that toning step is critical. You can pull out unbound silver. Yeah, you can, you're reducing that silver to a visible black. And again, overtoning will fade to a brown and fade to a red. And you have to watch this step microscopically. So you're going to have to start getting used to this toggling, okay? Don't overheat your silver, or you're going to flash your silver. You can't come back from that if you flash your silver. You have to start the whole procedure all over again. And if you overheat your silver, you're also going to crack that Copeland jar. And then you have a broken Copeland jar with flashed silver inside of a tub of inside of a glass tub that you're going to have to clean out. Then we have enzyme and immunofluorescence. So these are enzymatic procedures, mostly dealing with muscle diseases, and they are performed on frozen sectioning only. And that's using that liquid nitrogen snap freezing. If you want to see how that's done, I've embedded a link for you. So direct immunofluorescence, yeah, you're going to collect your frozen section slide, and then you're going to flood that slide with what's called blocking solution. It's usually a hydrogen peroxide solution. And what this does is it blocks the red blood cells from staining and interfering with the fluorescence that we're looking for. Yeah, then you're going to flood your slide with an antibody, that contains a fluorophore or a chromogen, if you will. Okay. But it is going to give the fluorophore. The difference between a chromogen and a fluorophore is that a chromogen will give light to color, where a fluorophore will fluoresce a color. Okay, so they are different, but they are both giving light to a color. The fluorophore is fluorescent. The chromogen is light microscopy. You're going to incubate in a you're going to incubate that fluorophore overnight. And then you are going to rinse it in PBS. PBS is phosphate buffered solution, and that's just going to rinse away your antibody. Yeah, then you're going to cover slip it and allow it to dry. But this guy has to be, it's really simple. Okay, it's simple. It's blocking antibody with a fluorophore, rinse it, cover slip it, look at it under a fluorescent scope. So this is kind of a neat diagram. Yeah, on this side is what's called indirect, and on this side it's direct. So the direct is the fluorescence, okay? The indirect is immunohistochemistry. So it's really kind of busted it down to easy, kind of an easy situation for you to wrap your head around. So if this is our cell, we're talking immunofluorescence now. So if this is our cell, and I want to, well, I'll do it when we do to indirect. So this is direct, done on a frozen section, okay? We haven't processed, we haven't fixed it. So outside here, this little yellow thing is your antigen, okay? So that's on the cell itself. They're your antigens on your cell. We then apply what's called a primary antibody. And so here's your fluorescent antibody. And on the tip, we have the fluorophore. So we've got a primary antibody with a fluorophore. So with the direct, you simply are putting these two things and attaching them to this. These are combined together in one solution, and you're attaching it to the antigen. Once you've attached it to the antigen, then you just hit it with saline to wash away the excess solution and cover slip it, okay? And then it will fluoresce. This is what's called an indirect method. And so here's our cell, here's our antigen, here's our primary antibody, and then we hit it with a secondary antibody, and multiple secondary antibodies can attach to it. And then we hit it with a fluorophore. So these are not combined, okay? These are all different steps. And the fluorophore in immunohistochemistry is actually a chromogen as opposed to a fluorescence. However, if you are doing you can do an indirect on a frozen section, okay? You can do a direct on a frozen section using a fluorophore for each. This, however, this indirect, if you are doing an immunohistochemistry, that means that you are doing a paraffin embedded section. And it's exactly the same with the antigen, the primary antibody, the secondary antibody. But with the paraffin embedded section, this would be a chromogen as opposed to a fluorophore. So again, let me repeat that. Both of these, using a fluorophore, both of these can be done on a frozen section. If you have a paraffin embedded section, you would do a indirect method using a chromogen instead of a fluorophore, okay? Because fluorophore fluoresces. Chromogen just brings light to light microscopy and can only be done on paraffin embedded tissues, okay? This is what's called the tau protein in brain. And this is an immunofluorescence. Yeah, so these little pink guys, or pink, I'm sorry, these little green guys are actually the tau that is fluorescing. If you, again, snap freezing, with immunofluorescence, you can snap freeze. I embedded a video into your frozen section, your other frozen, your general frozen section lecture. Yeah, and if you want to review it in your book, at least in my book, it's pages 286 to 288. And that's it, okay? Now remember, steps, functions, and results. steps, functions, and results. Now and forever. That's what you're gonna need to do. So you're gonna have to know what each one of those steps are. You're gonna have to know what each one of those steps do, and you're gonna have to know what the results are. Okay. So happy studying. Hope this did you well, and I'll see you next time. Bye.